You've probably seen in movies or read in science fiction novels, from The Matrix to Strange Days to Total Recall or The Sixth Day, how next-generation brain interfaces could work in the future. From recording and replaying extravagant vacations, to cures to epidemics, to downloading your college education in a few minutes. What they leave out is precisely how these technologies work. It is science fiction. The technology is taken for granted, and we see how these new technologies change what is possible. In order to get to the next level where this kind of technology is possible, we need to learn more about how the brain works at higher resolution and using new methods that have only recently been discovered, and where new developments are happening all the time. Areas like synthetic biology, nanoengineering, and human genome sequencing. Now is the time to make this a reality. The goal of brain backups is to do non-invasive, non-destructive imaging of brains. The current way to image brains at high resolution requires destroying the tissue by slicing it very thin. We think this is destructive and wasteful. Brain Backups is developing these tools to address medical and research problems in neurological disease, to understand memory formation, and map the brain at greater resolution without having to destroy the brain, or worse, kill the animal. And ultimately, to build a brain backup. Just like when you get a new iPhone, you can create a backup of your contacts and data on your computer or the cloud and then download them to your new phone. A brain backup can provide a backup of your synaptic connections that might one day be downloaded to a new brain or computer. The brain map of all the connections in your brain is called the connectome. Every cell in your body has a copy of your genome, but you only have one connectome, and it changes over time as you learn and age. Being able to measure the human connectome at multiple stages in a person's life will enable great new advances. We have been working on this for a long time, and the tools necessary to build this have become dramatically clearer in published literature, in industry developments, and in our own calculations. So far, we have meticulously designed functionalized DNA aptamer nanoparticles. We have worked on smart contrast agents for MRI and X-ray imaging, and DNA barcoding genome sequencing technology can be used to map neuronal connections. This fundraising campaign will allow us to do more groundbreaking research and get more data on how to map the brain and how the brain changes over time using these new imaging techniques. The campaign for brain backups will help sponsor new research and development toward making this a reality. This is a very exciting time for neuroscience research, and we want to share that with you. We sincerely appreciate your support and want to give back to you with some special rewards. Developing a map of the human brain without having to destroy that brain and being able to image it as it changes is a concern that is relevant to us all.